Hello, my name is Dominic Hondolero. I work at the NOAA Kasutsina Bay Lab in Seldovia, Alaska. Today I'm going to be talking to you about the prevalence of paralytic shellfish toxins in the marine food webs of coastal Alaska. This project has several collaborators from all around Alaska, as well as our NOAA lab in Beaufort, North Carolina. This is an NPRB project that was funded in 2019 and will continue through 2022. We have many sites around Alaska, as you can see from the map on the right. The objectives of this project were to develop saxitoxin screening and analysis capacity in Alaska, as well as to characterize Alexandrium blooms and phytoplankton blooms. Another objective was to examine toxins in zooplankton, forage fish, and then commercially important predatory fish. Particularly, we're concerned with the organs and muscle of those predatory fish. The reason that we're examining the organisms throughout the food web is to look at the risks to higher level food web consumers such as salmon, as well as human health and concerns for the seafood industry in Alaska. To collect the samples, the plankton samples are collected with surface samples from net tows. The forage fish samples are collected by beach seine, cast net, trawls, as well as stomach samples from predatory fish. And then the predatory fish samples are collected by sport catch, set net, as well as fish processors. And then we have two different methods of analyzing the toxins, both ELISA screening and HPLC screening. So what I'm going to be presenting first is Alexandrium cell abundance, and we have several years worth of data that are shown here in different colors going back to 2012. The first thing to note is that the y-axis is shown on a logarithmic scale, and what we're looking at is cell abundance, which is the number of cells per liter. As I said, the different years are shown in different colors. The red line is showing 500 cells per liter, and that is around the abundance of when we start being concerned about Alexandrium in the water, and we would start being concerned about toxicity and begin more intensive sampling. So as we're looking at this, we can see that there's been a general trend of increasing cell abundance for Alexandrium through 2016, and then that dropped down in 2017, 2018, and 2019. Next, we're going to be looking at our forage fish data. This is from 2019, so we had 249 samples that were analyzed. This is the toxicity, so this is shown on a different scale from the previous slide. So toxicity is shown in micrograms of toxin per 100 grams of tissue. And the scale of this graph goes up to 60. At around 80 or at 80 micrograms per 100 grams is when the FDA would say to stop consuming. So we can see that from these samples, these are the forage fish samples again, we can see that the toxicity was fairly low for all of these samples, except for a few very hot samples that we had. One Dolly Varden sample had an elevated level, as well as one of the Sandland samples. So these were the forage fish samples that were caught by beach seine. Next, we're looking here at the predatory fish samples. So these are 73 samples that were analyzed. From each of the fish, we collected digestive organs, liver, kidney, a muscle sample, roe if it was a female, and then we also collected the stomach contents, and each of those were analyzed separately. So we're presenting from the 4 different species of salmon here. We have pink salmon, sockeye salmon, Chinook salmon, and coho salmon. And again, all of these samples were below that FDA recommended limit of 80 micrograms of saxitoxin per 100 grams of tissue. There were a few samples that went over. If we're looking on the top right, we can see that one of the sockeye samples from the liver was above. And then in the bottom left, there was a high level of toxin in the Chinook kidney samples that we saw. Other than that, the toxin levels from all the predatory fish and all of the separate organs from those fish were very low. So to summarize, from our first year of data, what we're seeing is that toxins are present in plankton, juvenile forage fishes, as well as predatory fish. However, these are highly variable in time and space. There's a widespread low-level toxicity in fish, and this was commensurate with the low level of Alexandrian bloom intensity that we saw in 2019. However, we have seen other years where that bloom level is much higher. For the salmon, the paralytic shellfish toxins were primarily found in the digestive tract and excretory organs. They were very low in muscle meat and gonads. So what this means is that there's a low level of human risk. Most people are discarding the digestive tract and excretory organs and only keeping the muscle. So this also means that there's little economic threat to the seafood industry. However, this could represent a higher risk to predators that are consuming salmon whole, as they may be ingesting the toxins that are in those organs. So we have more questions. The field work will hopefully be continuing this year. Our questions are: what happens during an intense Alexandrian bloom, and what is worse, higher toxin levels versus a higher incidence of exposure to the toxin? And we would also like to know if there's an upper limit to the body burden of toxins, and whether or not at higher levels it can have any kind of effect on fishes. And that's it. If anybody has any questions about our research, feel free to contact me. If I don't know the answer, I can put you in contact with one of our other partners. And thank you for listening.